there, so we are on the problem solving questions, experimental design from the 2015 past paper, question 5. Okay, so our context, obviously the most important thing, read it through very carefully, make sure you understand exactly what it's talking about. So we have algal cells and photosynthesis in algal cells can be measured by immersing them in bicarbonate indicator solution. The indicator solution changes colour as the carbon dioxide is removed from it by photosynthesis. So it's just telling you everything. Colour change can be measured by placing the solution in a colorimeter. The higher the rate of photosynthesis, the higher the reading on the colorimeter. That's going to be important. The effect of different wavelengths of light on rate of photosynthesis in Cenedesnus, I can never say that properly, we're just going to call it algae from now on, which grows near the surface of fresh water lochs, was measured. The apparatus shown below was set up in a darkened room. Okay, so we've got our lamp. A light of a single wavelength because we've got a filter to allow only one specific wavelength to pass through, hitting a glass tube which has 50 megs of the algal cells immobilized into 10 gel beads, and then you're told that you have 5 centimeters cubed of 5% bicarbonate solution in there. Okay, there's your setup. After 1 hour, the bicarbonate indicator was removed, placed in a colorimeter, reading taken. The experiment was carried out 7 times using filters, each of which allowed different wavelengths of light to pass through. So we've got our different filters down here, 1 to 7, and the wavelengths of light that they actually passed through, and then how much the colorimeter reading was. Now remember, it's told you that the greater the reading, the greater the photosynthesis. So there's a lot of photosynthesis happening here. There's not very much happening here. Okay, so identify 2 variables not already mentioned that would have to be controlled to ensure that it was valid. Okay, so here is our data about the experiment, so let's look for things that we could talk about. You've not really got a lot of space left to talk about the bicarbonate indicator because you've got 5 centimeters cubed of 5%. That's pretty clear cut. The glass tube, you could talk about that. You want this to be exactly the same thickness of glass. You want it to be the same diameter so that you've got the same interaction between the bicarbonate indicator and the cells. You could talk about that one. You certainly could talk about the cells in terms of they should all be the same age, the same algal type. That's reasonable. And you could talk about the gel beads because you want them to be all exactly the same diameter, because otherwise you're going to have a different density of algae within each gel bead. 7 times using filters, I mean, all of this I think is reasonable, but where the lamp is, you're going to have to make sure that this is the same distance every time so that you get the same light intensity coming through. That's your main ones. A control tube would be required for each wavelength of light being investigated. Describe the contents of a suitable control tube. Okay, so we are looking at the concentration of, or sorry, the effect on the algae. So what we need to do is remove the algae as an issue. So what you would do is take this out somehow. So you basically still have gel beads but with no algae. Or what you might do is do the totally brutal biology thing, take the algae, boil them, cool them, and then still immobilize the same mass, because then you know that it's not anything to do just with the kind of chemistry of what's there. What you're looking at is a biological response. Okay. State why the tubes were left for 1 hour before the colorimeter readings were taken. Well, if you don't leave them for 1 hour, then you will not have any results, because you need enough time for the photosynthesis to occur and for you then to get results from the experiment. Okay, our next question is a line graph. You notice that I can't remember the last time you were asked to do a bar. This is higher. You're expecting to be drawing line graphs. Colorimeter readings against wavelengths of light. You are looking at a nice one in terms of they've actually given you wavelength of light in nanometers and the 400. So you now just need to work at a reasonable scale. Now if you look at this scale, if you go up in hundreds, you're going to be there too quickly. You're going to not use 50% of your graph paper reasonably, so you should be going up in 50s. Okay, up the side I need to get, our smallest number is 0.24, our biggest is 0.96. If I start at 0 and I go 0.2, 0.4, 0.6, 0.8 and 1, that will fit. I need to put this whole table heading over here, not missing anything out, putting the thing that says brackets units, close brackets. Okay, you need to have that in as well. And then we're looking for, so that, all of that, 1 mark. And then your second mark is for your plot being neat and joined neatly. Okay, remember, if anything is not on a point, you have to be able to get the half box tolerance. I think all of these will be on points. Yeah, okay. Okay, give the reason why the graph of colorimeter readings against wavelength of light can be described as an action spectrum. Now, this is actually a piece of content from the photosynthesis key area, but I've left it in to just see if you can see how it links in properly. So an action spectrum shows you how much photosynthesis is taking place. So if we have the wavelength of light, and then we have a number from the bicarbonate indicator color change, and that is linked to photosynthesis, that is then an action spectrum. Okay. Okay, the experiment was repeated with a second alga which lives in the water below the one that I'm going to pronounce incorrectly, so we'll just say the first alga. Predict the colorimeter reading the indicator would give after exposure of the second alga to light of 500 nanometers and explain your answer. Okay, so here's some kind of tricky logic, right? So at 500 nanometers, the first alga had quite a low colorimeter reading at 0.36, so it wasn't doing much of the photosynthesis in this range. Its high readings were up here and here. Okay, so this light at 450 and this light at 650 has almost all been used up, we can considering it that way, okay, by the first alga. This one has not all been used up. It's actually left some of this to kind of get past it into the kind of deeper water. So this one, I'm expecting it to be more than 0.36, anything more than 0.36, because what we're saying is this is it's taken out some light and it's left, what is left is what the other one's going to use. So you would expect a pattern that kind of mirrors the first pattern. So where the first pattern has had high photosynthesis, there'll be very little of that particular wavelength of light that's going to be available for the second one, so it will have low photosynthesis. But where you've got low, there's going to be lots of that light that's still available, and so therefore that's going to be used. And that's what you need to link up in your explanation. Here's our mark scheme. Quite a big mark scheme because there's lots of alternates in lots of different places. And that's another one done.